The endoplasmic reticulum, or ER, is an extensive membrane network that plays essential roles in protein and lipid metabolism. The endoplasmic reticulum is located throughout the cytoplasm and is continuous with the outer membrane of the nuclear envelope. Structurally, the ER consists of interconnected membrane-bound tubules and flattened sacs called cisternae, enclosing an internal space called the lumen. The endoplasmic reticulum occurs mainly in two forms: rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough ER appears rough because ribosomes are attached to its cytosolic surface. These ribosomes are responsible for synthesizing many proteins. Rough ER is a major site for synthesis of proteins destined for secretion, membranes, lysosomes, and parts of the endomembrane system. As translation occurs, many newly synthesized proteins are simultaneously transported through translocon channels into the ER lumen. Within the ER, proteins undergo folding and important modifications, including disphoid bond formation and initial carbohydrate modification. The ER also performs quality control, helping properly folded proteins proceed, while defective proteins are retained or targeted for degradation. Rough ER also produces transport vesicles that carry newly synthesized proteins from the ER toward the Golgi apparatus. During cell division, the ER and associated membranes contribute to the reassembly of the nuclear envelope around daughter nuclei. Cells specialized for protein secretion often contain extensive rough ER because they require very high rates of protein synthesis. Smooth ER lacks surface-bound ribosomes and is typically organized as an interconnected network of membrane tubules. Rough and smooth regions are parts of the same continuous ER system, and specialized smooth regions can arise through changes in ribosome association. One of the major functions of smooth ER is lipid synthesis, including the production of many phospholipids and other membrane lipids. In steroid-producing cells, smooth ER participates in the synthesis of steroid hormones, such as testosterone and estrogens. In liver cells, smooth ER contains detoxification enzymes, including cytochrome P450 systems, that chemically modify many drugs and harmful compounds. The ER also participates in glucose metabolism in liver cells, particularly in processing glucose-related intermediates during glycogen breakdown. A specialized form of smooth ER found in muscle cells is called the sarcoplasmic reticulum. Zooming into the sarcoplasmic reticulum, here, calcium ions are stored in high concentration. Voltage-gated channels pull open, where they will initiate muscle contraction. Zooming into the sarcoplasmic reticulum, here, calcium ions are stored in high concentration. Voltage-gated channels pull open, where they will initiate muscle contraction. The released calcium initiates molecular events that allow actin and myosin to interact, producing muscle contraction. After contraction, calcium is actively pumped back into the sarcoplasmic reticulum, allowing the muscle to relax. Rough ER mainly supports protein synthesis and processing, whereas smooth ER specializes in lipid metabolism, detoxification, and calcium handling. The ER works closely with the Golgi apparatus, forming an important pathway for modifying, sorting, and transporting cellular proteins. Thus, the ER is not simply a membrane network, it is a central hub connecting protein production, lipid metabolism, calcium signaling, and cellular transport. In summary, the endoplasmic reticulum is essential for protein production, lipid synthesis, detoxification, calcium regulation, and intracellular transport. Understanding the ER is understanding how eukaryotic cells manufacture, process, store, and transport their vital molecules.